Thank you all for being here. It's been a pleasure to hear from so many diverse scientists and now to be a part of the presenters. Today I'll be talking about SICM under concentration gradients, so I hope you all will remember what Lane and Martin were discussing. So I don't have to explain SICM, I don't think, but what do I mean by concentration gradient? It's just simply having one electrolyte inside my probe, which is the dark blue, and a different electrolyte outside my probe, which is kind of the light blue. And so why might we do this? In my case, it was to improve SNR. So if I want to keep my lateral resolution during SICM, I need my pipette to be as small as possible, and that places a restriction on my signal. And I can boost this signal without taking any other hits by just increasing the concentration of ions in my pipette. In addition, we can use this technique to probe surface charge properties, and I'll sort of ask you to take my word on this, but I'll also highlight some of Lane's other past work where they showed that the surface properties that you interrogate with your nanopipette can reflect in the current voltage relationship that you measure through the pipette. And so this is a very important concept for me. And then just to highlight another possible use, which we did hear about in biphasic SICM, you can increase your working distance. You can really improve the breadth of solvents that are accessible in your experiments through these techniques. And so in my case, I am trying to image neurons. I have this little panned SEM of a neuron culture, and so I can put numbers to my concentration gradient. Externally, I'll have a 140 millimolar physiological buffer, and then internally, for my pipette, I can go as high as about 3 molar. And when I was trying to do these measurements, I noticed something which was unexpected, and I'll gloss over why it was unexpected. But without a concentration gradient, the pipette essentially looked like a resistor, and with the concentration gradient, we then had this nonlinear current voltage transfer function through the tip. And I did some finite element modeling, and I showed that electroosmosis is responsible for this behavior. If I zoom in to the extreme points that I modeled, I found these really beautiful fluid vortices that develop at the tips of the nanopipette. So in these plots, the color, the brightness is the velocity magnitude of the electrolyte. The streamlines show you where it's going. So we get these really nice effects. And we published that in JPhys Chem A if you want to read more about it. And for the rest of this talk, I'll be presenting a little bit of new information and explaining why this is of interest to me and hopefully other people who are trying to use SICM for bio applications. So as we heard from Lane, a big thing is how can we use these probes to go beyond topographic imaging? And some work from them has shown, and it's in a little bit of a different paradigm than I'll talk about, but essentially your approach curve will depend on your voltage and it will depend on your surface functionalization. So in this top panel of figure A, at negative voltage with a positive surface, you get a regular curve. At positive voltage, you get this spike. If you invert the surface charge, now your inversion, so to speak, has also inverted. Sorry to use that word so many times. And we also see this effect goes away at high concentrations, which is due to the double layer effect. And a big thing is that we can use a concentration gradient to overcome this double layer limitation. And so they used this to show, with imaging that they could model the surface charge of these known materials. And I'll just quickly highlight that. Another group has done this, and suffice to say, their method is even more complicated. And it relies on a phase measurement between mechanical oscillation and the ensuing current. And there's interest in this. There's some methods to do it that are already shown. I'll be describing a slightly different method, which I'll just call surface charge mapping with hopping mode SICM. So we heard a little bit in hopping mode, SICM, at every pixel that we image, we start far away from the surface. We take an approach curve, which looks like this. This just depending on your pipette size. And we set some threshold value, and we can map our surface based on how close it requires to get before we hit this threshold. And so what I mainly am going to be showing in this talk is theoretical work that I've done with a finite element model. And so in these plots, which you'll see on the next slide, I just model my pipette as some long cone. It's suspended in some big electrolyte bath, and we have varying charge at the surface of the bath. So here I'm assuming, for example, that my biological sample is flat, or that it's very large compared to the opening of my tip, which is a decent assumption. And I apply a voltage to the inside of the pipette. I have a ground plane at the limit of the electrolyte bath. I have some inside concentration. I have some outside concentration. And the key variable is this distance between the tip and the surface. And in the finite element model, I can change my geometry, and then I can simulate what signal I'm going to see when I'm actually under the microscope and doing these experiments. And so first, let's focus on these middle plots, which would be in 140 millimolar buffer. This is for a positive this is for a negatively charged surface. This is for a positively charged surface. And the black and the red are different polarities of my pipette voltage. And so without a concentration gradient, we see that these curves mostly overlap. If I'm going to try and discern surface information, maybe I can do it. People may not believe me. Anyway, it's suspect. But now if we look here, if I have a 5x concentration difference where my external electrolyte is more concentrated than my internal electrolyte, we see this very useful divergence where as I approach the surface, if I have a positive voltage, we'll actually see an increase in current due to those surface-induced rectification effects that I highlighted earlier. And if we invert the concentration gradient, so it's again 5x, but now it's inside is greater than outside, this effect goes away with a negatively charged surface, but it's retained for a positively charged surface. And again, these are due to the way that electroosmotic flow and double layer effects interact in this thin limit where my transfer function through the pipette depends on the surface as I get close enough to it. And if I just change my internal kind of control concentration to 30 millimolar, we see pretty similar effects. Now, because we're in a lower ionic strength condition, these effects show up even without any concentration gradient. But in these instances, we can use the concentration gradient to, for example, switch the dependence of the approach curve on our bias, or even just to show across a range of conditions. If I predict 4 different results and then I show them across 4 different conditions, now I'm starting to actually convince my audience that what I'm talking about is what I see. And one thing to note is that these results I'm showing seem to be more sensitive to positive surfaces than negative surfaces. And that's not great for biology, which is normally negatively charged. But I hope you'll take my word for it. If we were to change the surface charge of our pipette, now we could basically switch all these relationships. And that's a pretty simple thing to do for people who work with pipettes all the time. And I'll conclude by showing some experimental data, which is also very preliminary, but highlights this effect. And so here I did SICM imaging of Pseudomonas aeruginosa with an NX Bio. And here we have my pipette tip. This is just a rough idea of how big I'm scanning, and all these little dots are the bacteria cells. And so I can highlight one trace and one image. And if I take the line scan, these circles are individual cells, the kind of smeared out shapes. They're just groups of cells. I didn't have enough resolution to discern. But anyway, we see about 1 to 1.5 micron protrusion from the surface due to these bacterial cells. And the really fascinating thing was that when I inverted my pipette voltage, I essentially got a negative of the image that I received. And so it's a little hard to see, but for example, there's a big cell here, corresponds to that. We can see this blob over there. And so I can show the line traces. This is a repeat from the previous slide. Here, you'll notice the dynamic range is only about 200 nanometers. And what was happening, much to my surprise, was that due to these effects, the SICM was recording the bacteria height as actually being lower than the flat surface. And that's really exciting because it's telling us that these things manifest in experiment, and it's telling us that we can learn about biology without really changing the imaging protocol too heavily or doing anything that complex. And I'll even note, despite the title of my talk, this was in the uniform concentration. I was hoping to flesh this work out a little bit more. Time did not permit. But anyway, I hope I've shown that the SICM signal will be influenced by the surface properties of your material. Changing the bias voltage allows us to interrogate these surface charge features without too much extra effort. Concentration gradients can amplify this effect and just prove the quantitative modeling that we've done. And we can do this with regular surface. We could do this with regular hopping mode imaging, and we don't have to modify any of the tools that our friends here work so hard to put in our labs. In the future, I hope to do more rigorous analysis of these experiments. And then really the bacteria are model cells, which are much easier to work with and understand than neurons. So ultimately, I want to use these same principles to look at the surface charge density that we would see at synapses. That's a big matter of interest in my lab. So I'll acknowledge my PI, who's not here. Martin's a collaborator. Ben and Jaili have been very helpful. Brian Choi, he used to help us out with the system. And that's about it.
Yeah. I saw one of your surfaces where the approach curve sort of went up. If you were to push that further towards the surface, is it dropping down again, like I think Lane showed earlier?
Yeah, it does. That data becomes not that useful or relevant to a real experiment. But yeah.
There's a paper where they did bilayers too, right?
I think so, yeah.
Yes. So the paper with bilayers where they see islands with different surface charge, which is probably pretty relevant to what you're.
Yeah, I should look at that more closely.
Yeah.
Thanks.
So what do you think is the reason for that negative profile when you change the voltage from positive to negative?
Right, so it's a little convoluted. I didn't do enough controls to characterize the effect of the bare surface. But essentially what happens if, suppose I have a strong negative charge on the surface of my bacteria, now when the pipette gets really close, there's extra ions over the surface of the bacteria. And that means my access resistance does not reduce as it does over the normal areas. And so my pipette has to come much closer to the surface before it reaches the threshold current. And the feedback loop doesn't understand why this effect happens. The feedback loop just raises the pipette and records a height based on where that threshold hits. So it kind of was touched upon by Martin indirectly, suppose I'm following this curve, and then eventually it does drop down. So now I've recorded a height which is much lower than I should have due to the effect of the surface.
If not, let's thank our